DNA is a nucleic acid that contains the genetic instructions of all living organisms. DNA builds things and is a long-term storage of information, made up of three groups: phosphate, sugar, and nitrogen base. Phosphate group is the same for all nucleotides. Phosphate always connects to the sugar. The bases, thymine, adenine, cytosine, and guanine, only bond T and A and C and G. You will never see a T with a C or G, or a C with an A or T. The phosphate is connected by a covalent bond. This bond is stronger and makes this the backbone, while the nitrogenous bases are connected by hydrogen bonds. A and T are connected by 2 hydrogen bonds, while C and G are connected by 3. A and T are easier to break apart than G and C. DNA is in a repeating pattern. Our model is laid out in a ladder form, but DNA is really put together in a double helix form. The process of multiplying the DNA is known as replication. Replication happens before cell division on the side of the chromosome called the origin of replication. This happens in the replication bubble. To replicate, the double helix has to be split and the base pairs part. Helicases travel to the origin of replication to unwind the DNA, forming a replication fork. Replication can only occur in one direction. The strands run anti-parallel, meaning the subunits on each DNA strand run opposite directions. Single-strand binding proteins hold the DNA apart so it doesn't immediately bond back together. Single-strand binding proteins hold the 2 template strands of the parental or original DNA apart. On the leading strand, the enzyme primase attaches to the DNA to synthesize and add an RNA primer. On the leading strand, nucleotides can be added continuously. The process is different for the lagging strand. First, on the lagging strand, primase removes the single-strand binding proteins. Then it forms an RNA primer. DNA polymerase III adds nucleotides to the 3' end of the RNA primer. These short segments are called Okazaki fragments. Finally, an enzyme called DNA ligase bonds the Okazaki fragments together so the DNA polymerase can run smoothly. DNA polymerase has to have the template from the original strand and then is proofread for mistakes or mutations in the DNA by the nucleotide excision repair when it replaces damaged DNA strands. This completes the lagging strand. The leading and lagging strands are then known as daughter DNA.